This meeting is being recorded.
So I think we should get started. I received a couple of emails from people that signed up for the webinar, but that will be a little bit later, but that's okay. Okay, so welcome everybody. So good morning, good evening, good afternoon, wherever you are in the world. My name is Matthew Vinken, and I'm the chair of the In2Tox Speciality Section of EUROTOX, which is one of the 5, as you probably know, specialty sections. So we specifically focus on in silico and in vitro toxicology. We started off in 2019 during the Helsinki conference. And yeah, unfortunately, of course, a couple of months later, we were faced already with the COVID situation, which for us has impeded really us of doing, what we wanted to do is to set up a lot of activities. But nevertheless, we don't allow the COVID situation to slow us down. So that's why we decided to have at least these, kind of webinars. Yeah, we had some webinars last year, and since the beginning of this year, we have a webinar like every month that is focused on at least one aspect that is relevant to in vitro and in silico toxicology. By the way, as you have heard at the start of this webinar, these webinars are being recorded, and you can actually watch them again on the EUROTOX website. So they are getting uploaded there routinely. I also want to advertise, of course, the upcoming EUROTOX conference, which will be in combined conference together with the International Toxicology Conference that will take place in Maastricht in the Netherlands from 18 to 21 of September. And actually, during those meetings, all of the specialty sections, so also In2Tox, get a slot to, also organize an activity, and that will be a meeting, a face-to-face meeting for a change. Where we will update you on our activities. So with this, I would like to introduce the speaker of today, who is Roos Masereeuw. She's a full professor of experimental pharmacology since 2015 and a scientific director since 2018 at Utrecht University for Pharmaceutical Sciences. She's trained as a pharmacologist and a toxicologist and obtained her PhD at Radboud University in Nijmegen, also in the Netherlands. And her research group focuses on understanding the pathways that can be pharmacologically triggered to enhance repair and regeneration after organ injury, with specific focus on the kidney. And for that purpose, her team is applying humanized in vitro systems that functionally mimic organs, and that include the use of innovative technologies like 3D systems and also organs on a chip. So just some housekeeping information here. So please mute yourself during the presentation. Also, please switch off your camera. In terms of Q&A, don't hesitate to send questions. I will keep track of this during the presentation in the chat. But of course, after the presentation, you will have your chance to also basically speak up and ask your question here. So with this, I will give the floor, the screen at least, to Roos. So thank you so much for accepting the invitation to present, Roos. We are really, really privileged to have you here. And please go ahead.
Thanks, Mathieu, for the kind introduction. I understand that everything is well visible, and I hope you can hear me too.
Excellent.
So,
all set.
Okay, thanks for confirming. Yeah, I'll be talking about kidney-based in vitro modeling, and especially related to drug-induced toxicity. And the content of this webinar is as follows. I will briefly recapitulate a bit the kidney function, kidney toxicity and biomarkers, then the translational challenges we are facing with respect to drug-induced toxicity screening, novel models, especially focusing on the novel in vitro models, and I will end with some take-home messages. So first of all, the kidney function. Well, obviously you're all aware of the important functions the kidneys have to the human body. They regulate the body fluid volume, blood pressure, and that's often the first sign that something is wrong with your kidney when blood pressure rises. Kidneys are also important for the secretion of some hormones like erythropoietin, but also the activation of vitamin D. So also patients that are suffering from kidney disease often have to take extra vitamin D because basically the kidney fails to activate the hormone and then all downstream comorbidities exist. Another important function is obviously the nutrient reabsorption, as the kidneys receive approximately 1/3 of the cardiac output, filters the blood, but with the blood filtrate, a lot of nutrients can be wasted. But fortunately, the tubular system of the nephrons is occupied with multiple transporter systems that are dedicated to reabsorb compounds like amino acids and glucose to maintain these nutrients for the human body. But the kidneys are also essential in the excretion of metabolic wastes and exogenous substances such as drugs. And this excretion is in fact a sum of different processes. And that's depicted over here. What you do see is blood is being filtered at the glomerulus. That's the first part of the nephron. The nephron is the functional unit of the kidneys. And then you have the tubular system, and the tubular system is a system that fine-tunes eventually the composition of the urine. Exogenous compounds like drugs or other potentially toxic compounds can be very well secreted by the kidneys and predominantly by the tubular cells. But it can also be absorbed more downstream the nephron segment. And finally, the renal excretion of a compound is determined by each of these three processes: filtration, tubular secretion, and tubular reabsorption. Now, in normal clinical practice, one way to monitor kidney function is by determining the GFR, the glomerular filtration rate. And this can be done by measuring an endogenous compound called creatinine. And it's basically a normal waste product. It's a product from muscle. It's a byproduct from muscle contractions, and basically there is a steady state level of creatinine in plasma. You can measure the creatinine in plasma, and when there is a severe rise in these levels, this means that the kidneys are not functioning properly. But it's also a very rough measure and doesn't really indicate any of the processes that can go wrong within the kidneys. Because if you do look carefully into the filtration rate, an increase in creatinine in plasma means a decrease in glomerular filtration rate. But this can occur by vascular effects, for example, renal vasoconstriction, by obstruction of the tubular system, for example, due to kidney stone formation, but also due to intrarenal factors, direct toxic injury either at the glomerular or at the tubular system. So you always need to do additional biomarkers to actually fine-tune where the toxic responses can take place. And you can look into the urine, where you can look into the reabsorption of amino acids and glucose that are predominantly being reabsorbed at the proximal tubules, and you can measure additional markers in urine, like, for example, kidney injury molecule 1, and then you know, well, indeed, the proximal tubule is affected. But still, it is difficult actually to fine-tune the specific mechanisms that are involved. And to do this in a proper way, you actually need to have a multiple number of test systems. But if you look into drug-induced toxicity, there is still a problem with the proper translation to the clinical situation. And this is nicely described by this paper by Gungrich in Drug Metabolism and Pharmacokinetics of 2011. He investigated actually the failure of drugs during development. And you can see that in preclinical tests, only less than 5% of the drugs that entered the pipeline were removed from this pipeline due to drug-induced kidney toxicity. But then further downstream in clinical trials, almost 25% of the drugs need to be removed from further development because of drug-induced toxicity. So you see a clear gap between the preclinical and the clinical tests. So this means that currently we do not predict well enough the drug safety in drug development. So this means we are facing really translational challenges. And there are two aspects due to these challenges. First of all, in vivo is not equal to in vitro. I already mentioned the complex organization of the kidney that's composed of glomerulus and a tubular system. You can see biomarkers like creatinine, an increase in creatinine in plasma, but how do we actually measure this increase in creatinine in plasma? How can we actually measure this in an in vitro system? So that there is really a challenge in translating in vitro to in vivo. And another challenge that we are facing is that animals are not humans. And this aspect I would like to demonstrate a little bit further to actually understand how well animals translate drug handling in vivo and translate to human drug clearance in vivo. We performed a systematic review. And in this systematic review, we investigated 20 different drugs that have a very clear renal drug clearance profile. And we looked into the kinetic aspects in humans, and we extracted the drug clearance values from animals from this literature. And then we actually used allometric scaling to estimate how well the animal studies predict renal drug clearance. And the allometric scaling is actually a scaling in which the renal drug clearance scales to the 3/4 power of body mass. This is a well accepted rule in currently applied preclinical studies for pharmacokinetics. And what we found was the following. So basically, we compared the renal drug clearance in animals and predicted the renal drug clearance in humans, and looked into the difference into which the predicted value actually compared to the actual value in humans. And the results are quite striking. Most animals do predict renal drug clearance quite well, except for rats, and that's depicted over here. We looked into all different aspects related to the 20 drugs that we accepted, that we included, and these belong to either anionic compounds, cationic compounds, or uncharged compounds, and all different physical chemical properties. And as you can appreciate quite well, indeed, rats do not predict renal drug clearing well enough. What you do see is that they overestimate secretion, whereas in dogs you see that they overestimate net filtration. But net filtration is not so much a risk factor as secretion is. So the main message of the systematic review was that rat models should be used with caution for drug disposition models. Unfortunately, rat is the most frequently used model in pharmacokinetic studies in vitro. So this is actually an important message, and we should look into this a little bit further. So I mentioned net secretion. is overestimated by rats. And if you have a clear look into net secretion, then this takes predominantly place at the level of cells of the proximal tubule. This is the first part of the tubular system, where cells are occupied with transporters that can handle compounds by taking them up from the blood compartment, transfer them via the tubular epithelial cells to be excreted into the urinary compartment. And you can see various different systems are present in these cells. For example, the organic cation transporter system. And for this, organic cation transporter 2 is an efficient transporter expressed in renal proximal tubules. This transporter system handles drugs like cimetidine, metformin, and cisplatin. And cisplatin I will introduce you in a bit further, but also creatinine. And you can see creatinine here. It's used as a glomerular filtration marker. Indeed, it is predominantly cleared by filtration, but to a small extent also by secretion. So this means that also whenever a drug is competing with transport for organic cation transporter 2, you can find an increase in creatinine. So you can also have a false positive increase in plasma creatinine, which does not necessarily reflect kidney toxicity. The third category of compounds being handled by the organic cation system is the class of so-called uremic toxins. These are endogenous waste products that accumulate in plasma in patients with kidney failure, and they can exert quite some toxic effects. But I would like to explain a little bit further cisplatin, because cisplatin is in vitro one of the most widely studied chemotherapeutic agents with a nephrotoxic potential. As said, it's a substrate for the organic cation transporter, but also for the copper transporter. So basically, it's taken up by two transporters in the kidney proximal tubules, and then it exerts a toxicity pathway which is quite complex and involves both mitochondria. It binds to DNA, and it uses a stress response, eventually leading to apoptosis at low doses, but at higher doses, it directly results in necrosis. So the first determinant is really the uptake of cisplatin into the tubular epithelial cells, because that's the major determinant of the intracellular concentration of cisplatin. And you can appreciate that cisplatin clearly shows a dose response curve. Here the cell viability is being measured at different time points, 24 hours, 48 hours, and 72. So it has a dose and time dependent character, which is indicative of a drug toxicant. Because it's a substrate of organic cation transporter 2 and the copper transporter, the toxicity could be attenuated by adding competitors for these transporters like cimetidine, which is a well-known substrate for the organic cation transporter and can compete with cisplatin, but also copper sulfate, which is handled by the copper transporter. Back to the tubular cells, so you can have uptake transporters at the side facing the bloodstream, but you also have transporters facing the urinary compartment. And for example, the mate transporters are important transporters and determinants for drug-induced toxicity, because they basically handle the efflux of compounds into urine. And you can see that the same type of substrates can be handled by these transporters. So how actually can we now better predict drug safety in now and currently and in the future? Well, most studies been done so far have been done in 2D cell cultures, which is quite nice to do. It's robust, but it doesn't predict drug safety in a human situation well enough. So we need better system, what we called advanced in vitro models. And looking into the literature of various models being used to study nephrotoxicity, you can see that still the majority of the models being applied are traditional 2D cell culture models. But you can also see, and here a validation score was developed to actually validate how well in vitro models that are being applied basically predict the human clinical performance. You can see that, apologies, it doesn't want to go back. Yes, apologies for this. So we actually applied the validation score and how well actually models predict the human situation and how well the studies were designed. You can see that advanced in vitro models, so more complex in vitro models, predict better than the traditional 2D models. But increasing complexity also comes with a price. It's probably costs more, most likely actually. We know that advanced in vitro models are also more elaborative to handle. So what type of advanced in vitro models are out there? Well, basically we categorize them in 4 different systems, and I will introduce them one by one and explain some advantages and disadvantages. And the first system I would like to introduce to you is the so-called Holofiber system. And that's a system that was developed in our lab by using polyethersylphone holofiber. And we actually developed the system to develop functional unit for the so-called bioartificial device, because we were interested in developing a better renal replacement therapy for patients with kidney failure. But this Holofiber system now is being used also in vitro in modeling kidney diseases and testing drug screenings. So the system is built of holofiber membranes that is double coated with an extracellular matrix, first L-DOPA, which functions as a natural glue. And for example, muscles also use this natural glue to adhere to rocks. So it's a very nice biological component to adhere cells to synthetic materials like polyethersylphone membranes. But on top of that, we also added collagen 4, which is part of the extracellular matrix in the normal kidneys. And if you then apply proximal tubular epithelial cells, then you can appreciate that these holofiber membranes are fully covered with cells, as is shown in this Z-stack image of the confocal microscope. And I'll play it again just to give you an indication. The cells are also nicely polarized, and here you can see the zona occludens 1 being stained in green, which is actually the tight junction proteins. In blue, you see a DAPI staining of the nuclei, and the organic cation transporter 2, which is facing the basolateral membrane of the kidney proximal tubules, is at the membrane, so at the basolateral membrane. So indeed, we could confirm the polarization of cells and also the viability of the cells in this system. Well, to demonstrate the functionality of the system, these fibers can be mounted in a chamber in which the fiber is mounted in between a perfusion inlet and a perfusion outlet. So we can mimic the blood stream by perfusing the fibers. And then if you perfuse the fiber with the fluorescent substrate, then you can measure with a confocal microscope how viable the cells are and how well they can take up this substrate. And we used in this example ASP, which is a fluorescent substrate for the organic cation transporter. And as you can appreciate over here in green, you see the uptake of ASP over time, indicating the cells indeed take up the fluorescent marker compound, which is a substrate for the organic cation transporter. And when you do this experiment in the presence of a mixture of uremic toxins, then you can clearly see a reduction of this transport. And also when we use cimetidine as a competitive inhibitor of this uptake, we see a diminished transport. We can test the barrier function because one important aspect of epithelial cells is that they form a tight barrier. And if you perfuse the fibers with fitzinolin as a marker molecule, then we don't want to see leakage from one compartment to the other, as shown here above in the 3 confocal images, where you see a clear leakage. So this is the experiment where the leakage experiment was done with fibers without cells, and you can see whenever you have a fully covered hollow fiber membrane, that the cells really form a barrier to fitzinolin leakage. And for your comparison, when the same cells are used in a transwell system, you can see that here the transwell leaks the fitzinolin whenever the transwell was used without cells, but with cells, you can see that there is only a minimal barrier. And that's what we often see with proximal tubular epithelial cells. They don't form a really tight barrier on transwells. But if you culture these same cells on hollow fiber membranes, you can clearly see a very strong barrier. So this indicates proximal tubular epithelial cells in a natural environment form a tube-like structures. And this is also what they like to do when you have them in vitro. You can use these hollow fiber membrane and fitzinolin leakage as a measure also to study drug-induced toxicity. And here, for example, we applied cisplatin at a quite high dosage, and you can see that the fibers indeed become leaky. And the same obviously holds true with the cell viability, which was clearly reduced after a short term exposure to cisplatin. But the hollow fiber membrane system can also be applied in studying pharmacokinetics. And in a study together with the group of Alexandra Galathin in Manchester, Tom van der Made actually used experimental data from the hollow fiber system, in which we studied the transepithelial transport of an endogenous molecule called inoxyl sulfate, and he could clearly translate this via a physiological based pharmacokinetic model to the clinical situation. So this means that the hollow fiber system is a nice system to study drug-induced toxicity, to study also drug handling, but it is a low throughput system, but with gain in mechanistic insight. So to come back to the systems, what other systems are there? Well, you can also use organoids. This is now currently being tested widely for all different purposes, for regenerative medicine purposes, but also for toxicity testing. And it was already discovered 20 years ago that whenever you grow kidney cells, and in this case MDCK cells, these are Medine-Darby canine kidney cells, if you culture them in a matrigel, so a stiff environment, they form spheroids, and they form clear polarized spheroids. Now this system is also being applied into a human system, and for example, you can use induced pluripotent stem cells, and if you grow them into a matrigel under specific growth factor conditions, they form kidney organoids. And you can use these kidney organoids, for example, also for cisplatin-induced toxicity testing over time, as has been described in the study by Grebinger. But you can also use organoids for more personalized medicine approach. And for this, the group of Hans Klevers, together with Marianne Verhaar, a nephrologist at University Medical Center in Utrecht, developed a very new platform called tubuloids. They actually isolated adult human kidney stem cells from urine that they grow into matrigel under specific culture conditions that also form these spheroids. And these spheroids are called tubuloids because they basically reflect the tubular system of the kidney. So unlike induced pluripotent stem cells, that whenever you grow them into kidney organoids can give rise to all segments of the kidneys, these adult stem cell derived organoids only give rise to the tubular system of the kidneys. But you can use these tubuloids also to scale up and use them as kind of factories for tubular epithelial cells that you can then isolate, and you can also use them to grow on holofiber membranes, as Rafael Lindoso did in our lab. And he could clearly demonstrate that tubuloid-derived epithelial cells form a nice monolayer on these holofiber membranes, and they also show a clear polarization and form a tight membrane barrier. The third system that I would like to present is a membrane-based microfluidics system. And it's basically a system where the cells are divided from two different channels that can be perfused and form a barrier in between these two channels. And this is a system that's first developed in Donald Ingber's group at the Wyss Institute, where they basically use a primary human proximal tubular epithelial cells to grow on a semi-permeable membrane and expose them to fluid streams at two sites. And what they could demonstrate is that flow really matters. If you would compare static culture conditions with flow conditions, then you can appreciate that the cell height increases. Well, they used human proximal tubular epithelial cells, and we know in normal life that these cells form cuboidal structures. So the increase in height is also an indication of a better differentiation of these tubular epithelial cells. And you see also an increase in transporters, and they measured the sodium potassium ATPase, an important ATP pump, another pump is, is the aquaporin 1, a water channel, and they could see that the number of ciliated cells also increases. And this is an indication that the cells indeed sense the flow, because cilia are the flow sensors of epithelial cells, and whenever you expose cells to flow, you not only see the increase in the number of ciliated cells, but you also see the increase in ciliary length. And this is important for downstream signaling. And what the group of Donald Ingber did in the next set of experiments is test also cisplatin-induced nephrotoxicity. And also here you see a striking difference in response whenever cells are cultured in static conditions or under flow. You can see a much higher increase in LDH under static conditions as compared to flow. But you see an overestimation of the toxicity effect in static conditions compared to flow. And what you also could demonstrate is that whenever you inhibit the uptake of cisplatin in the tubular cell, under flow conditions, you can completely reverse the toxicity, whereas in static conditions, this is not the case. On the other hand, you could still see that the cells are sensitive to apoptosis whenever the experiment was done and compared between static and flow conditions. And you see no difference in response to apoptosis, whether or not there is flow, but you see again a clear reduction in the response when the inhibitor was tested. Well, overall, the conclusion was of this study that the experiments under flow much better predict the clinical situation than the static conditions. Well, the same system was also used to develop a so-called human glomerulus on a chip. And with this same chip model, they could model adriamycin-induced glomerular injury. And they could demonstrate that especially the podocytes, which are the epithelial cells that form the slit barriers in the glomerular basement membrane, that these cells are sensitive to adriamycin, which in fact in clinical situation is also the case. And in addition, I would also like briefly demonstrate the system that was developed by Mark Zhuo's group called Tissues, which is a membrane-based system where cells are cultured in a transwell setup, but connected via flow. And in this system, the group could demonstrate that you can also combine multiple organs with each other. And in this case, they combined liver spheroids with kidney proximal tubular cells to study the cyclosporine A-induced toxicity. And they actually did a repeated dose study in which the kidney proximal tubular cells and the liver spheroids were exposed to cyclosporine A for 14 days. And they looked into the toxicity on both organ systems, alone or in combination with rifampicin. And they could clearly demonstrate toxicity in both organ systems, but they could also demonstrate that the addition of rifampicin decreased the cyclosporine-induced toxicity in the kidney cells. And this has all to do with the fact that rifampicin can reduce the uptake of the toxic compound in the kidney cells. So in this way, the membrane-based microfluidics system can be used to study also longer-term drug toxicity effects. Well, this is actually a summary of the 3 systems I have presented so far. The gel-based system, which basically gives tissues an organoid or organization, a holofiber membrane where we can perfuse the system and study transepithelial transport processes of a membrane-based microfluidics where cells are still cultured on a flat membrane but exposed to flow at 2 sites. Well, they all 3 could clearly demonstrate that they have an in vivo-like environment with improved morphology, but they are still quite complex systems and not really compatible with high throughput analysis. And to overcome this, I would like to introduce a 4th system, which is basically a more hybrid system. It's a gel-based system, but also in which microfluidics is applied. And the system is what I would like to present is the so-called Organoplate, developed by Mimeta's company in the Netherlands, that has a very unique approach. They developed a microtiter plate in which, in fact, 96 kidney on chip or organ on chip systems are compiled. And this microtiter plate is composed of these small functional units, where a ECM channel is being used to form an extracellular matrix. In this case, collagen is being used, and the collagen is used for cells that are perfused in a perfusion channel to adhere to. So the construction of this channel is such a way that the cells can adhere to the extracellular matrix at the phase guide and then form a tube-like structure as shown here. And by perfusing the other channel with medium, they can mimic the bloodstream, and the perfusing the cells in the medium channel can mimic the luminal compartments. So in this way, you can both mimic the urinary and the bloodstream compartment in one system. It's a Wells plate, and this Wells plate can be placed on a rocker platform, and then the shear stress is introduced by forming fluid flow based on gravity. So basically, a rocker platform is responsible for the shear stress. And then you can, for example, measure barrier function, like we did also with the Holofiber membrane. You can use a fluorescent marker compound and measure the leakage of the marker compound from one compartment to the other. And the cells should form a leak-tight barrier. But whenever cells are disrupted, you can see that the fluorescent marker can leak to the other compartment. So the platform is actually a very elegant platform that's compatible with a higher throughput if you would combine it with a fluorescent microscopy. But you can also sample the perfusion medium and do transepithelial measurements, or you can do some qPCR marker biomarker analysis. To demonstrate how actually this can be applied for studying, for example, transporter interaction, this experiment is very elegant, because in this way, P-glycoprotein mediated transport was demonstrated in the organoplate. Well, P-glycoprotein is an efflux pump, is also often a drug target, and the following experiment was done. Calcium AM is a substrate for P-glycoprotein, and whenever you perfuse the tubes within the organoplate with calcium AM, you won't see much, because P-glycoprotein is so efficient in pumping the calcium AM out of the cell. But whenever you do this experiment with an inhibitor of this pump, like for example the PSCA33, which is a experimental compound, then you would see that the calcium AM is retained in the cells and can be converted by esterases in the cell into the fluorescent calcium. And that's shown over here. So in this case, the cells were perfused with calcium AM in presence of a blocker of P-glycoprotein. The calcium AM is retained in the cells, converted into calcium, and that's what you see over here. So in this way, you can study drug interactions on a specific transporter. But you can also measure the mitochondrial activity by, for example, doing a WST assay, or you can measure the lactate heat dehydrogenase as a measurement of membrane integrity loss or cell viability loss. And this is what we did in an experiment where we exposed these organoplates to 2 concentrations of cisplatin, tenofovir, another well-known nephrotoxic agent, tobramycin, and cyclosporin A, and you could clearly see that the cell viability was compromised at higher doses for cisplatin, tenofovir, and cyclosporin A, and that you also see an increase in LDH release. Finally, the organoplate is also compatible with using the adult stem cell-derived tubuloids, as was nicely described in a Nature Protocols of last year by Linda Gijzen and Fyodor Yevgeny, who demonstrated that these tubuloids form indeed very nice tube-like structure and can be applied for drug screening processes on a higher throughput way. So this brings me to the end of this webinar, and obviously in a minute it will be open for questions, and I will address the specific questions you might have. But let me first give you some take-home messages. In fact, there is not an ideal kidney-based in vitro model to study nephrotoxicity, so you should really select the model based on your research question. If you want to do a drug screening on a wider basis, then you should have a high throughput system. But this high throughput system doesn't give you necessarily the true mechanistic insights you want to have. So if you want to have a true mechanistic insight, you will go to a more membrane-based system or the Holofiber system that is more compatible with a larger analysis of the data. But I do believe that microphysiological systems have the potential for a better prediction of drug performances. And I think eventually that advanced in vitro models might lead to safer drugs. And maybe we should combine the system in the near future directly with in silico models to have the true translational value. And with this, I would like to thank the audience for listening. I'm grateful to the many sponsors of the work that I just demonstrated that was also sponsored by different agencies, the European Union, the Dutch Kidney Foundation, and NC3Rs, for example. And I'm grateful to many people with whom I collaborated over the past few years within my group at University Utrecht, together with the University Medical Center in Utrecht, Radboud UMC, and Mimetas. And with this, I would like to end my presentation. This is now open for questions. And I would like to remind you that the next In2Tox webinar is scheduled for Tuesday, the 19th of April, in which Mathieu Vinken, the chair of today, is actually presenting liver-based in vitro modeling.
Yeah, thank you so much, Roos. This was an excellent talk. I'm also impressed by the many papers and the high-end journals you guys have. So I think you're indeed the right person to, give this kind of webinar. So thank you so much. Are there any questions from the audience? So you can either speak up, but then you have to unmute, obviously, or you can share this in the chats for people that are a bit more shy, so to say. Well, maybe to trigger discussion a bit, I can ask a first question. So Roos, a major problem that is now faced these days in not only, I assume, in the kidney prediction or the kidney adverse effects prediction field, but also for other organs, or so-called idiosyncratic reactions, so that are very difficult to predict. So to what extent do you think that the models that you have presented are able to predict such an effect?
Yeah, very good question, Mathieu. That's difficult. So I think for direct toxicity, it's not so difficult. The models I demonstrated, idiosyncratic often involves the immune system. So I think if we want to predict idiosyncratic drug reactions, I think we need an immune component. And I think that this is really essential. And this holds true for all organ systems, in fact, where you see the idiosyncratic reactions occur. And I think that maybe the liver is more sensitive even than the kidney is. But also there it holds true that you need a better immune system. And so far, the immune system on a chip has not been developed, as far as I know, well enough to do this. The other problem with idiosyncratic drug reactions is that they are so rare. So how, in the many measurements you do, how will you pick up this idiosyncratic effects? I think that here artificial intelligence might also help. Whenever you have a pile of data, you should actually use artificial intelligence. You should use an unbiased way of measuring all parameters that you have measured to kind of predict whether you can expect an unexpected event. Yeah.
Okay.
But I think we're not there yet. But it is definitely something that should be addressed in the near future. And I think this is something that you need that needs a multi-component approach.
Absolutely, and also an interdisciplinary approach. So in the meanwhile, we got 2 questions in the chat. I will just read out loud. So how can you mimic GFR effects of drugs?
Yeah, basically you can mimic by this is done by basically mimicking a filter. So the GFR effect can be GFR. The filtration rate is based on size and a little bit of charge, so that it's quite clear how selective the filter is. If you're really interested in toxicity effects on the filter, then you should build a glomerulus on a chip. And that's something you can do by using podocytes that form processes, food processes that are the barrier slits and that can be done on an artificial membrane. So same as what I demonstrated for the tubular system, you can use a semi-permeable membrane and then use podocytes to form the slit pores. So you can mimic filtration rate.
Okay, thank you, Roos. Then another question, but I do not know if I fully understand. I think the question is, what would be the most reliable and human relevant model of all of those that you presented today? Yeah, I see the question too. Yeah, the best model doesn't exist. I think a model is always a model. You need to understand its boundaries to apply it in a sensible way. I like organoids because they approach human physiology very good, but they're also immature. You must understand that the kidney organoids are basically at the fetal status of a kidney, and drug transporters and metabolism enzymes actually mature even after birth. So we're not there yet having a adult kidney model in vitro yet. So we can also only model parts. So the model we have with the Holofiber system is a mature tubular model, but it obviously has some complications because it's very elaborative. It's not high throughput, so you have to compromise there.
So again, with you should select your model based on your research question, and yeah, the most optimal model will hopefully be developed soon.
Okay, thank you. Maybe a final question from my side, because in the interest of time, I think we should wrap up in a couple of minutes. But what, according to you, will it take to get all of these, or at least one of these models or systems accepted by the regulators? Because in the end, you want to use this in contemporary, let's say, testing in pharmaceutical settings, and they have to meet, like regulatory guidelines. So what would be your take on this?
Yeah, the best approach is to really demonstrate the predictability. So for each model to be applied, you should demonstrate the predictivity, and that's the most important thing. We have to find the evidence that the model predict well enough. And therefore, I also like the combination of having in vitro experimental with in silico, because you need the scalability. You need to demonstrate to what level the experimental findings relate to the human kidney situation. And so I would say approach the regulators with a package of in vitro and in silico. And only then I think it makes sense. So a model by itself, I don't think will get the full approval. It's really the application.
Absolutely, and a change in mindset, basically. But there's a lot of work. Okay, so with this, I think we came to the end of the webinar. So first and foremost, of course, I would like to thank Roos for this excellent talk. As you know, well, this has been recorded. So for the participants, of course, I also like to thank the participants for attending. You can watch again this webinar. Please have a look at the website of EUROTOX. And last but not least, I would like to thank, of course, also Elaine. She's a bit hidden here behind the screen, but she's taking care of all of the logistics and the practicality. So that's really, really appreciated. And with this, I also would like to invite you to the next webinar. So it will be up to me then next month to talk about liver-based in vitro modeling. And again, I cannot emphasize it enough, but please consider also attending the EUROTOX and the International Conference on Toxicology meeting in Maastricht in September. So with this, thank you so much, everybody, and have a good continuation of your day. Bye.
Bye-bye.